afternoon, students. Today I am back with the second part of anatomy of flowering plants. Okay, so today we are going to start with anatomy of dicotyledonous root. We are going to start with anatomy of dicotyledonous root, and this is a TS of a dicotyledonous root. So let us start with the first point. First point is what? The outermost layer is called epiblema. The outermost layer of a root is called as epiblema. Many cells of epiblema protrude in the form of unicellular root hairs. Please remember that root hairs are unicellular. Why I am telling you this? Because it is very important for your NEET MCQ, and why it is so important that we will come to know later in this chapter. Okay, so many of the cells of epiblema protrude in the form of unicellular root hairs. The cortex consists of what? After epiblema there is cortex. After epiblema epidermis there is cortex. Cortex is made up of what? Cortex is made up of thin parenchymatous cells. These are the parenchymatous cells. These are the parenchymatous cells, and here we can see intercellular spaces. Here we can see intercellular spaces. Okay, so cortex consists of parenchymatous cells which has intercellular spaces, but generally see this epidermis epiblema region does not have intercellular spaces. So this is till cortex. Now what is the innermost layer of cortex? This one, this pink one, this is the innermost layer of cortex, and it is known as endodermis. The innermost layer is known as endodermis, where there is a single layer of barrel shaped cells. See, here there is a single layer of barrel shaped cells, and these are again without any intercellular spaces. These are again without any intercellular spaces. Why such? Because it prevents water seepage into the inner vascular bundle. It prevents water seepage into the inner vascular bundle. Now what? So this part we are done. Tangential and radial walls of endodermal cells have a deposition of water impermeable waxy material. This wall surrounding the endodermis has a water impermeable thick cuticle with a waxy material suberin, and they are known as Casparian strips. They are known as Casparian strips. Now pericycle is present just next to epidermis. Pericycle is present, this one, just next to epidermis. Sorry, endodermis. Sorry, my mistake. Pericycle is present just next to endodermis. Let us start from here. Okay, so pericycle is this one. Pericycle is present just, if this is the endodermis, this is the pericycle. If that is the endodermis, this is the pericycle, and here we are having 2 to 4 xylem and phloem patches. Xylem and phloem patches. Like this. So, towards the radius, these smaller ones are known as protoxylem. Everything is written here. Everything what I am telling is actually written here. Okay. I am just explaining to you now. These smaller ones are known as protoxylem. These bigger ones are known as metaxylem. Okay. And these cells just beside xylem is known as phloem. These cells just beside xylem are known as phloem. Cambium ring develops between the xylem and phloem. Here we can see formation of cambium between xylem and phloem. That means this is a open vascular system. Now, how will we connect our previous day's class and this class? This is the trick for qualifying NEET. Open vascular system means what? First is it has cambium. Second point was it can make secondary growths. It can make secondary growths. Okay. So, and we know that, and where this is present? In dicotyledonous systems. We knew that open vascular system is mostly present in dicotyledonous plant, and this is also root of dicotyledonous plant. And we know another thing, that from the morphology chapter we know something, that taproot is present mostly in the dicotyledonous plant. We know that taproot is present mostly in the dicotyledonous plant. And what taproot has? It has secondary roots. It has secondary roots. That means secondary growths are there, and secondary growth is possible only from open vascular system. This we learned from the first part of this chapter. ठीक है बच्चों, कुछ याद आ रहा है, कुछ समझ में आ रहा है कि the secondary growth is present in dicotyledonous plant. Again, cambium is present in dicotyledonous plant, and secondary growth is again possible only through open vascular system or where cambium is present. So abhi we can connect. Okay, now we will connect and study. Like each and every chapter is not segregated, they are not discrete, they are all interconnected, so we have to understand that way. Okay, now there is a term pericycle plus vascular bundles plus pith is known as stele. Pericycle plus vascular bundle plus pith is called stele. Acha, another thing is that pith is small here. Pith is small here in comparison to the monocotyledonous plant. So this is another point that we have to remember. So now let us go to monocotyledonous root. Anatomy of monocot root. It is quite similar to dicot root. That is, it is also having epidermis, cortex, endodermis, pericycle, vascular bundle and pith. But there are few differences. Here there is no secondary growth. Here there are no secondary growth. Because what I told? Because here there is no open vascular system. Here there is no open vascular system. And usually there are around 6 or more than 6 xylem bundles. See? Rather than 2 to 4 in the dicot. Here there are more xylem bundles. Correct? In dicot there were only 4. Here there are 6 to 7. So this is another difference. Okay? Pith is large. Here this pith is large. In case of dicots, pith was small. And monocots do not undergo any secondary growth unlike the dicot roots. So these are the few differences. Otherwise, epidermis, cortex, endodermis, everything is same as that of the dicot. Okay, number of xylem phloem are more, no secondary growth, and mostly it is a close vascular bundle, and pith is large, Okay, so these are the few things that we have to remember. So now let us come to anatomy of dicotyledonous shoot. This is the cross section, this is the cross section, and this is the transverse section. Okay, we will come to this picture in details later on. So this is the cross section of anatomy of dicotyledonous root. Here, epidermis is the outermost protective layer. Here, epidermis, this one. Epidermis is the outermost protective layer of the shoot. And epidermis is covered with cuticle, bare trichomes or epidermal hair, and a few stomata. Stems do have stomata. Okay, few stomata, fewer than the leaves, but it does have. So there is epidermis. From epidermis rise epidermal hairs or trichomes, and there are few stomata. Now, why I told you to remember that root hairs are unicellular? Because here trichomes are multicellular. Trichomes are multicellular. Now, cortex here are arranged in multiple layers. Let me just this view off. Cortex is formed of multiple layers between epidermis and pericycle. Outer one is known as hypodermis. Outer part of cortex is known as hypodermis. This is this layer. This layer. This is hypodermis. Okay. What is hypodermis? It consists of a few layers of collenchymatous cells. This one. It consists of a few layers of collenchymatous cells just below the epidermis. So this is just below the epidermis. यहाँ पे देखो. This is just below the epidermis. Below this consists of rounded, thin-walled parenchymatous cells. Rounded, thin-walled parenchymatous cells. It is this one here and this green part here. This green part here. Okay, this is the second layer of cortex. This is the second layer of cortex. Now let us come to the innermost layer or endodermis. This one is the innermost layer or endodermis, and just below this is the pericycle. Just below this is the pericycle. Here, if you want to see this thin one, this thin undulated thing, this is endodermis and this one is pericycle. This semilunar thing is a pericycle. This one is pericycle, and this thin strip, this is endodermis. And what is the special characteristics of endodermis here? It is, the special characteristics of endodermis is it is rich in starch grains. It is rich in starch grains and also known as starch sheath. Achha, by the way, if you want these notes, such crisp notes of NCERT, these are like complete coverage of NCERT. So if you want such notes, then you can contact me in my WhatsApp number, which is given in the description box, and I can share the notes with you. Okay, so let us come back to the chapter. Now, after this endodermis lies the pericycle. After this endodermis lies the pericycle. Pericycle is present on the inner side of the endodermis as semilunar patches of sclerenchyma. This is the pericycle, like a semilunar patch, and this is sclerenchymatous cells. These are sclerenchymatous cells. Now comes phloem and xylem. We can find that here there is a conjoint type of vascular bundle. In stems there is a conjoint type of vascular bundle. You remember the first part of this chapter? I told you about radial arrangement like this, conjoint arrangement like this. So this is the conjoint arrangement. Here is the phloem, below is the xylem. Here metaxylem is towards the periphery and inside is the protoxylem. And between phloem and xylem is cambium. Between phloem and xylem is cambium. Clear? Now, what are medullary rays? There is another term called medullary rays. These are few layers of radially placed parenchymatous cells in between the vascular bundles. These are radially arranged parenchymatous cells between the vascular bundles, and these are known as medullary rays. These are known as medullary rays. These are this part. It's clear? These are the medullary rays. This is phloem, these are xylem, this is cambium, and these are medullary rays. These are medullary rays. Okay. Now, a large number of vascular bundles are arranged in a ring. A large number of vascular bundles are arranged in a ring. Each vascular bundle are conjoint, open, and endark. Endark means protoxylem is towards inside, protoxylem is towards inside, and metaxylem is outside. This bigger metaxylem is outside and protoxylem is inside. Now each vascular bundle, अब इसमें confuse नहीं होना है, each vascular bundle is conjoint and each vascular bundle is arranged in a ring pattern. Each vascular bundle is arranged in a ring pattern. Okay. So इसमें confuse नहीं होना है. This is not radially arranged this thing, vascular bundle. This is a conjoint. But this each vascular bundle are arranged in a ring pattern. Now clear? So this is all about dicotyledonous shoot. Now let us come to monocotyledonous stem. Here again presence of sclerenchymatous hypodermis. Here there is a presence of sclerenchymatous hypodermis. I mean, these are the few differences that we have from the dicot stem. These are the few differences we are having from the dicot stem. Large number of scattered vascular bundles. There were large number of vascular bundle but arranged in a ring. Here they are scattered. Here they are scattered and surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. These scattered vascular bundles are covered by what? Surrounded by sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. So these parts are covered with these things. Vascular bundles here are conjoint and closed. Mostly in case of monocot we will find closed. In case of dicot it is open. In case of monocot it is closed. Clear? See, these are those sclerenchymatous cells. These are the parenchymatous cells. These are the large conspicuous parenchymatous cells. Peripheral vascular bundles are generally smaller than the central located ones. See here, this is a cross section like a ring. See, the peripheral ones are smaller than the inside ones. Peripheral ones are smaller than the inside ones. Phloem parenchyma is absent and water containing cavities are present within the vascular bundles. We can see here that water containing cavities are present in the vascular bundles. Phloem parenchyma is absent. Okay, so these are the few most important characteristics of monocotyledonous stems. Now we will be coming to dicotyledonous leaf. Again, the main parts are epidermis, mesophyll and vascular system. Epidermis, mesophyll and vascular system. Mesophyll is also again divided into two parts that we will be coming later. Okay, so this is the epidermis. Epidermis is of two types here. Adaxial means that is present at the top of the leaf. Adaxial is top of the leaf and abaxial is below the leaf. Okay, abaxial is below the leaf. So here the epidermis is of two types and generally stoma is present where? Stoma is present at the back. Stoma is present at the back of the leaf, that is the abaxial epidermis. Epidermis of the leaf has a conspicuous cuticle. So that we know, epidermis of leaf has a conspicuous cuticle because we know that leaf has to minimize its transpiration. So it has cuticle. Abaxial epidermis generally bears more stomata than adaxial epidermis, and adaxial sometimes even may lack stomata. So this point I have already covered. Now mesophyll, the tissue between the upper means adaxial and lower, that means abaxial epidermis is known as mesophyll. Mesophyll is again divided into two, palisade and spongy. Palisade and spongy. What is the main function of mesophyll? Mesophyll consists of chloroplasts. The reason for which leaf is so important to us and to the whole ecosystem, that responsible part is this mesophyll. It carries out photosynthesis. Mesophyll carries out photosynthesis. Now it has again two types of cells, palisade and spongy. So this oval ones, these are palisade mesophylls. This oval ones are palisade mesophylls. And this round ones, see, it has air cavity between it. They also have little bit of intercellular space, but not such huge air cavities. So these are known as spongy mesophyll and these are known as palisade mesophyll. These are known as spongy mesophyll and these are known as palisade mesophyll. Okay, so what are the few characteristics? It is situated just below the palisade cells. The spongy ones are situated just below the palisade cells. It extends to the lower epidermis. Numerous large spaces and air cavities. Now comes vascular system. Here it includes vascular bundles and seen in the veins and the midrib. The vascular bundles are mainly seen here in the veins and the midrib, and also the size of this vascular bundle, this part. This actually runs through the vein. This runs through the vein and the midrib of the plants, and size of this vascular bundle depends on the size of the veins or the size of the midrib. Now veins vary in thickness in reticulate venation. We know that a small leaf might have a smaller vein and midrib, but a bigger leaf will have larger one. So based on that, this vascular bundle shape and size will also change. Okay, vascular bundles are surrounded by layer of thick-walled bundle sheath cells. They are surrounded by thick-walled bundle sheath cells. You cannot see this, no? Wait. These are the thick-walled bundle sheath cells. Okay. So this is anatomy of dicotyledonous leaf. Now let us come to monocot leaf. These are isobilateral leaf. By the way, in case of dicot, it is dorsiventral. Means we can see the upper part is more green, the lower part is lesser green, like that. And isobilateral means it is almost similar from both the surfaces. So in general, monocot has isobilateral leaf, but anatomy is much similar to the dorsiventral leaf. Anatomy is much similar to the dorsiventral leaf. In an isobilateral leaf, the stomata are present on both the surfaces. Although I know here the picture shows stomata only here, but in oh no, it's also here. It's not marked. See, here it is also there. Okay, so stomata is present both in adaxial and in abaxial epidermis. Okay, mesophyll is not differentiated into palisade and spongy. Mesophyll is not differentiated into palisade and spongy. So this is the mesophyll. Okay. Bulliform. These are modified adaxial epidermal cells along the veins which forms large empty colorless cells. Bulliform is actually a specialized type of epidermal cells which runs along the veins and which forms large empty colorless cells. This. Okay, now when bulliform cells in the leaves have absorbed water, they are turgid and the leaf surface is exposed, and when they are flaccid due to water stress, they make the leaves curl inward to minimize water loss. So these are the bulliform cells, and when they are turgid, the leaf is straight. The leaf is straight. Sorry, this has become too much straight. Anyways, the leaf is straight. Okay, so these are parallel venations. Now when this bulliform leaves are flaccid, what happens? The leaves curl. The leaves curl. So less exposure to the atmosphere. So less exposure to the atmosphere. See, the total surface area here is more than the total surface area here that is exposed. Obviously, we can understand that. And parallel venation in monocot leaves result into almost similar sizes of vascular bundle. As in reticulate venation, we saw that the size of the veins are different. Midrib is thicker and larger, and this is larger than this vein. This is larger than this vein. But in case of monocot or in case of parallel venation, we have seen almost similar size of veins. So which makes the vascular bundle also of similar size, which makes the vascular bundle also of similar size, except in the main vein. This main vein is a little bit larger than the other leaves. Okay, so till now we are done with anatomy of monocotyledonous leaf, and for this class, thank you, happy learning, bye bye. And if you like my channel, then do subscribe my channel. If you want notes, then you can comment me, and also you can text me in the WhatsApp number given in the description box. So bye bye.